So, good evening, everyone, ladies and gents. Welcome to the KPPC, the Kuwait Public Policy Center. As you're used to our flagship program, whereby we hold the lecture series on a monthly basis, this is a special edition that will be a webinar. Today, we have a special guest, Dr. Farley, which I'll go into a bit more details with regards to his background. Just a quick agenda. We'll have a brief statement from His Excellency, the Secretary General of the Supreme Council, Dr. Khalid Mehdi. Then we'll dive into the program of tonight. So, a quick background about Dr. Farley. Dr. Farley is an international expert who has over 30 years' experience in international health development, research, and program implementation. As an infectious disease thought leader, systems thinker, and epidemiologist with particular focus on HIV and AIDS, he works at Palladium. Dr. Farley leads the global health practice, an interdisciplinary team in 4 markets: US, Europe, Middle East, Australia, Pacific. He focuses on holistic responses to global health priorities based on sustainable health systems, implementation science, health promotion, and prevention. He has had a distinguished career at the US National Institute of Health before serving as a senior scientist and faculty member at the University of Maryland Medical Center. He holds an MD and an MMPH from Johns Hopkins University and is trained in internal medicine and infectious diseases. So just a quick housekeeping for those that are joining right now. Can you kindly mute your computers, please? Right now, we'll have a brief statement from His Excellency Dr. Khalid Mehdi, the Secretary General of the Supreme Council for Planning and Development. The floor is yours, Your Excellency.
Thank you very much, Faris, for organizing, and the whole team from KPPC organizing the Kuwait Public Policy lecture series. Given the time that we are going through, we are not stopping our activity. We are working as usual, as well as we are even innovating different and many ways and expanding our activities. This is our first virtual lecture of Kuwait Public Policy Center lecture series, and we hope to continue doing it monthly, the usual, as if we are working in our offices and premises. I would like first to welcome Dr. Farley Cleghorn, which I had the chance to meet virtually. We didn't have a chance to meet in a face-to-face meeting, but I would say we already met. We had a very interesting discussion throughout a very important project that carried by the KPPC with Palladium Group. I was very impressed with the team and the whole work that's been done, especially when it comes to managing the COVID-19. I would like to welcome you, Dr. Farley, for being with us today. It is an honor to listen to you and learn from your experience and expertise with regard to the COVID-19 as well as any other pandemic. Over to you, Faris.
Thank you very much, Dr. Khalid.
And thank you, Your Excellency. Thank you, Your Excellency. Now we'll be moving on to Dr. Farley. Just once again, a reminder to all those joining this conference, kindly mute your computer, please. Dr. Farley, we're honored to have you today as part of the KPPC flagship lecture series program. The floor is yours, please.
Thank you, Faris, and thank you, Dr. Khalid, for inviting me to give this important talk and to have this important discussion. As you've heard, my name is Dr. Farley Cleghorn. I am the Global Head of Health Practice at Palladium, and I think many of you know Palladium. We are an international consultancy group, and we have a large health practice that works all over the world. And one of the incredible opportunities that we have had in order to make an impact in the world is, of course, to assist countries with their coronavirus epidemic responses. And this is something that is happening all over the world, and it is happening in real time. And one of the important ideas to remember is that for many people, this seems to be something new. But as I will discuss with you over the next hour, this is something that we have been predicting for quite a long time. Some of the details are slightly different, but it's not new to people like me who've been working in infectious diseases for a long time. I would like to say that it's impossible with all of the information that is emerging on this particular pandemic for us to cover all of the data that is being generated around the world. And I think it's something for us to remember that we're generating probably more information about this one disease than we have done on all diseases combined in such a relatively short time. The challenge for us will be to convert this information into usable knowledge. And that usable knowledge is to be used in service of improving our responses to the epidemic, to reduce the mortality associated with the disease, and of course the morbidity, that is the illness itself, and to reduce the impact of the pandemic on society. In fact, the biggest impact of pandemics is disruption to society. And that is why in managing the response, we've always got to take a balanced approach to understanding what we should be doing based on biomedical information and what will lead to the most efficient recovery and return to some notion of normalcy. So in this talk, I have many sections. Throughout the talk, I will be presenting concepts or ideas that will help you to understand the story of the pandemic. And when you have a good notion of the story of the pandemic, it helps you to put into perspective detailed information that you will get about any part of the response or any part of the science. It helps you to place these within context. So that is my objective today, is to give you the story of the coronavirus pandemic and to think about Kuwait as an example of a country that is one of every country in the world going through some stage of mitigating the impact of coronavirus. I'll start off by talking about where we are today. I'm sure you're all using various apps and various websites in order to get updated information on the pandemic. And the way that we generally report this information is we look at country reporting. We think about the numbers of cases that have been confirmed from countries around the world. And up to today, over 6 million cases have been reported, and over 370,000 deaths have been reported. This, of course, has happened over the last 3.5 months. We'll talk about the timeline of the epidemic shortly, but the notion that you start off with a new disease unknown to humanity, described in China in December of 2019. And here we are at the end of May, 5 months later, we've had over 6 million cases and 370,000 deaths. Keep in mind that the website I'm using here, by the way, is based out of Oxford University. I chose a neutral country, so to speak, to give you the update. Every country in the world is represented, including Kuwait. And we can talk about what we mean by reported cases, what we mean by a COVID-19 death later on in the talk. But if we assume that the numbers are correct, they are generally an undercount of the number of cases, an undercount, because reporting takes time in many places and there are barriers to accurate reporting, one of them being getting enough testing done. And one of the criteria for a reported case is having a positive test. If you just take the fraction of 370,000 over 6 million, you will get around 6%. We will talk about mortality rates later on in the talk, but keep in mind that the true mortality rate for the epidemic is likely to be lower than 6% because we aren't testing enough people around the world to know the true extent of the epidemic. One of the rules of thumb that we as epidemiologists use is to take the number of deaths to establish a mortality rate, let's say of 1%. 1%, by the way, would be 10 times more than the seasonal flu epidemics. So at a rate of 1% with 370,000 deaths, which as we said is an undercount, that would imply that we have 37 million cases circulating in the globe, and we have detected 6 million of those cases with testing. That's just a rule of thumb for you to remember. Now, having said that is where we are today, what is the history of this pandemic? We mentioned earlier that it's only been 5 months, but it's been a very detailed history. First of all, let's talk about what we mean by a pandemic. Pandemic is when an infectious disease is circulating in multiple places around the world at the same time. Now, you'll see slightly different definitions for this in various places. The WHO definition says circulating on at least 2 continents. We know that coronavirus is circulating everywhere in the world today. There isn't a country that has not reported some circulation. There are many differences, however, by country, and we'll talk about those later. So what are the prerequisites for a pandemic? The first is that we must have the emergence of a novel agent. We refer to infectious disease-causing organisms, sometimes as agents. And so a novel agent emerges to which the population has no immunity. And that becomes important later on when we talk about vaccines and we talk about testing. The second is this new agent must be able to replicate and cause disease in humans. In other words, if this coronavirus caused no discernible effect, it would be unimportant. But the fact is that this coronavirus causes, as we just saw, significant mortality, 10 times greater than seasonal flu. And it happens in such a short time scale that it has overwhelmed health systems, even in the most advanced countries. Thirdly, the new agent must be transmitted effectively from person to person. One of the, for those of you that remember the early days of this, I can recall on January 11th, I gave a talk on coronaviruses to Palladium in Washington, D.C. At that time, we had 400 cases in China. And we were just beginning to see cases in Korea and Japan. And someone asked me, well, transmission, what do we know about transmission? And I said, well, if it's based on our experience with SARS or with MERS, and we'll talk about those 2 infections later on. If it's based on our experience, we found that asymptomatic people did not transmit the virus very effectively. And therefore, that's a good thing. If it's like the flu, I said, which is transmitted quite efficiently between people who are not symptomatic, then we have a big problem. As it turns out, the latter is the case, that coronavirus is very efficiently transmitted between people who are sick and those who are not sick. So that is the prerequisite for a pandemic. And have we seen this before? We have seen it many, many times. And I'll just, on this slide, give you some notion historically of epidemics and pandemics that occurred in the last 2,000 years. Many of you have heard of plague, you've heard of smallpox, you've heard of the Spanish flu of 1918, and most of the comparisons that you will see to coronavirus are to the Spanish flu epidemic of 1918. Of course, the Spanish flu epidemic is misnamed because it actually first started in Kansas in the US. But because people were returning from World War I, it was dubbed the Spanish flu. We can talk some more about that later. In the last 50 years, we've seen multiple agents that have done the jump from animals into humans. Some of you will know that the annual flu epidemic, which happens every year, is actually a good representation of a new emerging organism that changes every year, and that's why we have a new epidemic every year. Some organisms do not change, and so we do not have annual epidemics. I'll explain this a little bit more later on. More relevant to the region, more recently, you've had the MERS epidemic, Middle East Respiratory Syndrome, which emerged in 2012. And this is also caused by a coronavirus. So in fact, we have many, many, many examples of diseases that have jumped from animal species to humans and which have attained epidemic potential, but we have not had one that has attained pandemic potential in this time that coronavirus COVID-19 has done. Many of you will remember the swine flu epidemic of 2009. In fact, a lot of people don't remember it at all because they never heard of it. But that also achieved pandemic status. However, it was a disease of much less importance because it caused much lower morbidity and mortality. Now, this is the same slide with some of the notions of morbidity and mortality on the slide. And it shows that for Spanish flu, which was 100 years ago, over 500 million people were infected and over 50 million deaths were recorded. That's an enormous number of people, given the fact that the world population has increased by a number of billion over the last 100 years. So this is really the most apt comparison. If we think about MERS, for example, Middle East Respiratory Syndrome, which is still going on, by the way, in Saudi Arabia, we've only had about 900 deaths and about 3,000 cases. So clearly, that is much harder to transmit than the coronavirus that is causing COVID-19. By the way, I just wanted to give a note about these slides. Some of these slides are quite busy, which you can read at your leisure later on. The majority of the slides, unlike our consulting slides, really try to portray a single concept that is helping you with this understanding of the coronavirus epidemic. So the concept on this slide is we've been here before, we have lessons from the past, and we can apply those lessons to the present state and to the future. This gives you some notion of diseases you might think are either under control or not a problem in your country, but they are problems in other countries. And again, it's a chronology of diseases by name that are also zoonoses, What is a zoonosis? A zoonosis is an organism that jumps from animal species into humans and causes some kind of disease. Now, many of you don't think of HIV as a zoonosis. In fact, HIV is a classical example of a zoonosis that jumped into humans sometime around between 1910 and 1920. We did not discover this disease until 1980, because often when diseases jump over into humans, they go unrecognized for long periods before they achieve what we call epidemic potential. And then when attention is turned to them, we realize this is something completely new. So in fact, when I think about the coronavirus epidemic today, I think less about the flu epidemic of 1918. I think about the last 30 years of the HIV epidemic. The difference between HIV and coronavirus is the time scale is very compressed for coronavirus, whereas the time scale for HIV is classically slower. But if you look at the mortality caused by the HIV epidemic around the world in the last 30 years, over 30 million people have died of HIV. So again, the concept that I want you to retain is that we have many, many examples of organisms that jump from animals into humans. There are conditions that allow this to happen. And one of the infectious diseases preoccupations is to understand what are those conditions. How do we know when this will happen? How do we predict how it will happen? And how do we prepare for it? So I'll leave you to read this at your leisure, but knowing that we have much, much experience to draw on. This slide is merely a heat map that relates where we expect organisms to jump from animals into humans. All parts of the world are potentially susceptible to this based on the conditions, as I said, that are variable around the world. The places that traditionally have been the hotspots, which are notated in red on this heat map, are the places where there are lots of wild animals. There are opportunities for contact between wild animals and people, and there are conditions among people that allow for the spread of those agents. As I said, this is happening every day. Most times it achieves not very much. You have a couple of cases that may go undetected and everyone goes back to normal. When we have transmissions or zoonoses that occur between animals and people that lead to societal impact, that tends to be the types of viruses like coronavirus that are very easy to transmit by standard human activity, such as social interaction. I'll mention again that every year we have this happening for the influenza epidemic. Every year we try to predict which strains of influenza will undergo what we call recombination in parts of the world like southern China, exactly where the coronavirus started. And the reason that this happens for influenza is that you have the confluence of people, animals, in this case, pigs and ducks for influenza, and you have the conditions for spread, which is overcrowding and lots of social interaction. So coronavirus is doing something that we go through every year with influenza. Now, when a novel agent, in this case, the coronavirus, emerges from an animal reservoir. Now, what do we mean by an animal reservoir? If we were to search, as we have been doing for the last many years, search for different types of new viruses in animal species, you will find that bats harbor more viruses than most mammals. And their anatomy, physiology, and systems have evolved to coexist with many of these viruses. We don't really know why that is so. Bats have an immune system that seems to be unbothered by many viruses that are deadly to humans. For those of you who recall the Ebola epidemic of 2014-2015 in West Africa, the more recent Ebola epidemic in Central Africa, both of these are thought to be seeded by bats. Similarly, when you think about SARS, a coronavirus, which was described in 2004, 2005, this also was well described in that bats, a particular species of bat, interacted with a particular mammal called a civet. Those civets were offered for sale in markets in China, which led to the crossover or spillover event into humans. And by the way, the spillover event is not about consuming the animal. The spillover event is about butchering the animal. It is butchering that allows the spillover event to take place. So what I'm saying to you is that bats harbor many significant viruses to humans, and we believe that this is exactly what has happened in coronavirus, COVID-19. We actually call the virus itself SARS-CoV-2, and we call the disease that it causes COVID-19. So keep in mind that we are pretty clear that this is the source animal for coronavirus. What we are not so clear about, and you'll see a lot of discussion of this in the press, did this happen in the wet market in Wuhan? We still don't have clear proof that that is the case. We know that the location of the initial outbreak was Wuhan. And so based on our experience with SARS, we were looking for likely places for the virus to emerge. But again, let me state that we do not have conclusive proof that the wet market is where this happened. Now, what happens after the spillover event? Well, you have to have the ability then to transmit between humans. And there are many modes of transmission between humans. For example, we mentioned HIV earlier. Well, the most efficient way to transmit HIV between humans is close sexual contact. The most efficient way to transmit coronavirus is through respiratory secretions. And so similar to SARS, similar to MERS, however, with much greater potential spread, if we compare the transmissibility of COVID-19, SARS-CoV-2, to influenza, it looks to be as infectious as the most infectious influenza virus. So again, thinking about how the epidemic starts, the spillover event must occur, the conditions for spread must be present, and for respiratory spread, this is the most significant and fast mode of spread that we have among humans. There's only one other mode of spread that would be more efficient than respiratory spread, and that is simple, what we call airborne spread. Airborne spread means that you're not even in a place where someone is coughing, but rather someone coughed there before you got there, and the virus hangs around in the atmosphere. That is airborne. And we are still debating whether for this coronavirus, there is significant airborne transmission. It seems likely that some of that does occur in closed environments. Now, we are talking a lot about coronaviruses. And I think many of you probably never thought about a coronavirus before. And you think about viruses in ways that they are, of course, very small organisms they're actually organisms smaller than viruses. But generally, viruses are considered to be on the sort of margin between living cellular organisms with a nucleus and genetic material and something else entirely. Some people have even debated if viruses are truly alive. But as you can tell from this pandemic, it seems to be quite clearly alive. This picture is a cartoon that is the official picture used by the CDC in the US. It actually is a model, and it shows the proteins that are on the spikes of the virus. This is why it was called coronavirus, because when we first saw the electron microscopic images of the virus, it looked like the virus was wearing a crown. By the way, this happened in 1965. This is when coronaviruses were first described in the medical literature. So quite a long time ago. And so based on this scanning electron microscope picture, this is why the name was given. Coronaviruses were described by a female virologist in the UK. And what does this represent? Well, if you look at this cartoon, you'll see that it's really a collection of proteins. And inside, you have the nucleocapsid protein, which is an RNA. You will hear many descriptions of viruses, but you can lump them into two categories: RNA viruses and DNA viruses. Coronavirus is an RNA virus. And so you've got that genetic material inside. The virus actually has to spread by attaching to human cells and then emptying their genetic material into the cell, which then takes over the genetic machinery of the cell to produce more viruses. This is how viruses behave. And they're unable to reproduce without entering a cell. The clinical effects, that is the illness, is produced when so many cells are taken over that the systems that support our daily life in the body break down. So a virus, therefore, like the coronavirus, is simply a collection of proteins, one of which is the nuclear protein, which allows it to reproduce itself. And later on, when we talk about testing and vaccines, understanding the structure of the virus is very important because it allows us to say, well, maybe if I looked at the spike protein and I found an antibody to the spike protein, I can make an effective vaccine. So structure is very important for us to understand with viruses. Many of you will, when we talk about the seasonal flu, will not have realized that part of the seasonal flu mix that we go through every year actually is represented by 4 circulating non-novel or common coronaviruses. So if you think about the common cold, and we do have tests for the common cold, you can actually identify 4 common coronaviruses that are part of what we call the common cold mix every year. What we are talking about today is not those coronaviruses. Those are the ones that have been described since 1965. There are 3 novel ones. We mentioned them all already. The first was SARS. SARS, as far as we can tell, is disappeared from the surface of the planet. Can we say that for a fact? Well, we would have to go back to the origin species, to those bats, and try to find a SARS-like virus. And we can do that, and we will find it. But there is no transmission of SARS among humans at the end of that epidemic. Similarly, MERS, which, if we think about species that harbor viruses, we normally would not think about camels as one of them. But as it turned out, camels do harbor MERS. And how camels got MERS in the first place is a source of conjecture and research, but it's very likely that camels also got MERS from bats. Now, MERS actually is now endemic within certain populations of camels in the world. And in Saudi Arabia, where I have worked, this is an ongoing problem. And there are still small numbers of cases that are occurring every year in Saudi Arabia from MERS. However, MERS has never achieved what we call epidemic potential. So even within the country in which it is described, the numbers of cases remain relatively low. And now today we have COVID-19, which, as we said, is called SARS-CoV-2, the actual virus. This is the official name of the virus in the WHO Taxonomy Committee. And the properties here are very different from SARS or MERS. It's very influenza-like, but it is a coronavirus. Let's give you a little more detail about these three, because it's very important for us to understand. You know, people think of this as something new, how did this happen? As I said, we have two recent examples of how this happened, one of which is SARS, the other is MERS. If we take the column on the left-hand side, you will see that, and we'll go through this in more detail a little later. First reported case in Wuhan, 2019. We said that the spillover event very likely took place through an intermediate species from bats. Now, we have not conclusively proven that this intermediate species was a pangolin, but you will see many news stories that refer to pangolins as the intermediate species. We don't know that. And most likely it was from butchering, as we said. We moved very quickly from the first cases in December of 2019 to having a global pandemic declared on March 11, 2020. And there have been actually over 6 million cases and 370,000 deaths. The current pandemic is ongoing. The future is unclear. However, based on our experience, we can model the future, and based on reality in the present, we can try to understand the pathways into a better future that controls coronavirus. So if we look at both MERS and SARS, as we said, limited spread. SARS has disappeared from humans completely. MERS continues to be an issue, but at low scale in one or two countries. The most affected country for MERS is Saudi Arabia. And it's likely that MERS will not spread to the rest of the world because it hasn't done so in the last 7 years. Occasionally, a case will spread from direct contact in Saudi Arabia with a patient to another country. That has happened in Korea, it's happened in other places, but it never goes beyond that. There's no pandemic potential for MERS. Now, we are now talking about this pandemic. We talked about the origin. Let's go into a little more detail about that origin. As you can see from this slide, the cluster of cases that was described in December of 2019 were initially described as atypical pneumonia. And this is a very important point because one of the ways that we can look at proxies for coronavirus cases in countries is to look for spikes in atypical pneumonia and pneumonia causing death that have no diagnosis. And that's something that we are still doing today in many countries. So a cluster of atypical pneumonia, and it was described as atypical because they didn't know the cause, in Wuhan, China. This led to reporting by January 31st, of the first fatal case. And January 7th, I'm sorry, that was January 11th. January 7th, the virus was isolated. It was not hard to do. It was literally take a swab from a patient of one of these cases, look in the lab, try to grow the virus using methods we have refined over 50 years. And it was very successful. Immediately, the virus was discovered on January 7th. And that once the virus was discovered, it was reported to WHO then. The sequence was shared with WHO, which then continued to share it with research groups around the world. And the tests for the virus were started, development of the tests were started right away, which is why WHO had a test very quickly for the virus. And then from that initial spread in Wuhan, China, and the first death on January 11th, immediately spread started occurring in other parts of China and in other parts of the world that had close contact with China. Now, what does that mean? Let me give you one fact. Between the initial report of coronavirus in Wuhan and the lockdown in Wuhan, an analysis done by the New York Times looked at every flight coming in and out of China in that period. And over 6 million passengers flew out of China in that period. And this is one of the lessons that we have to understand, that when you think about lockdowns, they have to be applied when they will have the maximum impact. So one of the initial lost opportunities in Wuhan was in the 3-week period between reporting the first death and the lockdown. A huge number of people were able to take the virus out of China and seed it around the world. Now, I'm talking about regular commercial flights, okay? Regular commercial flights. It just goes to show how enormous our commercial flight infrastructure is around the world and how impacted it's been by the epidemic. We learned a couple more things in January of 2020. One is that cases were being reported from other places. The other is that healthcare workers could become infected quite easily. And secondly, that the virus and the epidemic didn't show the same features necessarily when introduced from one country to another. We were beginning to see differences in how the epidemic emerged. Now, we said that it took until March 11th to declare a global pandemic. We defined what a pandemic is. It is a spread occurring on at least 2 continents in the world. We actually, in the US, for example, had the first death from COVID-19 on February 29th. And this was a direct link, we think, to Wuhan. Every country will have a different, what I call, origin story. Essentially, from Wuhan, we had seeding to many countries in the world, one of which was northern Italy. The northern Italy epidemic actually seeded the New York epidemic. And we can do this by looking at what we call the phylogenetics of the virus. We can sequence the virus and look for small changes that mark what we call lineages. We can look at where did the virus in point A, did it come from B or C? We can actually build up that map of the spread of the virus around the world. So the epidemic in New York was seeded from Italy. The epidemic on the West Coast of America was seeded from China. So a different time scale. I mentioned 6.5 million people flying out of Wuhan before lockdown. None of them flew out of Wuhan to carry the virus. They were all going home or going about their business. This is also a type of heat map that shows commercial aviation flights around the world. And if you think about, I mentioned SARS earlier on and said that SARS has disappeared from the planet. SARS started in 2004. If I showed you this heat map from 2004, it would be 1/8 of what commercial aviation is in 2019, or was actually, because commercial aviation has been one of the early victims of the pandemic. 98% drop in commercial aviation. So keep that in mind. A huge density of commercial flights shrink the world, allowing for spread. If the spread is of a very infectious virus, this easily accounts for the way that the virus has spread around the world, easily accounts for it. This is one of the conditions for dissemination of the virus. When you think about infectious diseases, on my slide number 2, I show that in the last 50 years, we've had more opportunities for infectious diseases to cross over from animals to humans. One of the reasons is we have shrunk the world. You can be exposed one day in Wuhan, and the next day you're home in California. Likewise, other conditions that impact spread include density of cities. One of the covariates that we have found for spread is the density of human populations and the use of public transportation. So 55% of the global population lives in dense urban settlements. And that is something that promotes spread of a pandemic like coronavirus. The other and third important point about the frequency of crossover events or spillover events from animals to humans is our impact on the natural environment. We are much closer to animals today because humans have expanded all over the world. We are building settlements in places where we didn't before. And then, as I said, the ease of transportation and travel allows that to impact everywhere around the world. I want to talk a little bit about how these epidemics develop. I showed you first a slide of the number of cases around the world. This is the same slide, actually. It shows that even within 5 months, we have a very shifting epidemiological picture. We mentioned that Wuhan is where this pandemic started. Well, they have had no new cases for the last couple of weeks. And the force of the epidemic, this is how an epidemiologist would describe it. The force of the epidemic has shifted around the world multiple times since Wuhan. And you would recall the epidemic in Italy, in France, in Spain, and within Europe, that has now shifted to the UK. Belgium has had the highest mortality rate in Europe. We can talk about that. Then, of course, the epidemic in the US. The US is actually too big physically to talk about a single epidemic. There are multiple epidemics within the US happening at the same time. Some of them are mature, like New York, and we're actually starting to see mortality go down in New York, but we're seeing mortality go up in other states that are just starting their epidemic. So when you think about this picture, if you think about the right of the graph, you will see that Brazil, in terms of number of reported cases, is about to overtake the US as the most affected country in the world. You will see that India is starting to rise up as well. And countries like in Europe, like the United Kingdom, continue to maintain a high rate. And at the same time, you'll see countries like Australia and New Zealand who have managed to actually significantly control the epidemic and achieve epidemic control. And there are valuable lessons from how they were able to do that. Every country in the world is represented in this website, which, as we said earlier, is run out of Oxford. And it's a source of very good information. Likewise, there are websites at Johns Hopkins University, my alma mater, and a couple of other very well researched and reliable websites that give you up-to-date information. And it's an interesting point to be made that all of the reliable websites are in the private sector or in the academic sector. We have seen that governments like our CDC in the US, their website for information is actually less reliable because they just don't have the resources to keep up with the changes. Now, if we think about if this slide shows reported cases, what is being reported? Well, unless you have a good case definition for an illness, you really can't compare countries. And so a lot of time and effort has been spent trying to understand what should be reported and to diminish the differences between reported numbers so that you can compare and combine countries. So essentially, what we have for COVID-19 is a set of criteria. The criteria include clinical criteria, epidemiological criteria, diagnostic imaging criteria, and lab criteria. And you build the case definition based on these four components. So the clinical criteria are what the patient is experiencing, those are called symptoms, and what the medical workers pick up, those are called signs. We look at a combination of signs and symptoms in order to define the clinical illness. Clinical illness alone is not enough to confirm a case. You need some additional evidence. Now, in many places, they've been looking at epidemiological evidence. That is, were you in a place that has established coronavirus? Did you have contact with a person who has an established coronavirus? That's considered epidemiological evidence. We use that evidence within a defined time period, which is the incubation period of the illness, which has a maximum of 14 days as far as we know. So we use that number. We don't use the lower bound of the incubation period, which could be 2 to 3 days. We use the 14 days to capture as many as we can. Similarly, you need some confirmatory evidence that we are talking about coronavirus here. And the most important one is to have a positive lab test. What test are we referring to? The test is to detect the presence of the virus itself. And that is an important distinction from detecting antibodies to the virus, which is part of your immune response to the virus. And we'll explore that in more detail. But ideally, for all reported cases of coronavirus, COVID-19, we should have a positive test that has detected a virus in the patient. And then finally, there are other criteria like X-rays that show classical pictures that I'll show you a picture shortly. It is what we call a ground glass reticulation in the lung. And this is quite typical of coronavirus. Now, in order to put these four categories of criteria together, we talk about possible cases. So if someone just has clinical symptoms, we can call that a possible case. And many countries, because they have difficulties in implementing testing, they have been reporting clinical cases. We have probable cases where we have two sets of criteria at least, and then we have confirmed cases, and that is where we have established the presence of the virus with a virus test. Now, when we talk about reported cases, there's another very clear marker that can be reported, which is death. Keep in mind that the natural history of the virus is exposure, infection, illness, outcome. What is the outcome? One of them is death. And death is, in epidemiology, we say death is easy to define because this makes it easy to report. When deaths happen very quickly, however, it can also overwhelm systems. And so many deaths can go unreported. But in general, if the system is not overwhelmed, death is easy to report. So it gives you a picture of the epidemic, but it's what we call a trailing picture, because death occurs on average 3 to 4 weeks after infection. So it's an important component of defining the epidemic. And on this graph, you can see, also taken from the same source, that some countries experienced a rapid spike and then a reduction. Some countries have had sustained deaths, and some countries are still to experience their peak in death. And I will congratulate you in Kuwait at this point, because you have managed to keep your mortality rate very low. I'm looking at the numbers for Kuwait today. It says 27,000 cases at least, 212 deaths. That is very low. Now, we can explain why that is, or try to explore why that is in the discussion. Here is a picture of the US deaths. This is overall US deaths in the epidemic until today, from February 29th. We're using a 7-day rolling average. We do that because we smooth out daily differences in reporting. And you can see the trajectory is in the right place, that the number of deaths are going down from 2,000 average a day to 1,000. That's still a lot of deaths, but the trajectory is in the right place. However, as we mentioned earlier, the US is actually too large to look at a single picture. You actually have to look at this picture for New York, and this picture for California, and this picture for Montana, and you will see that they're all in very different stages of their epidemic. Now, a very important point, and this could explain the Kuwait numbers, as we'll discuss later, is that there's a very clear relationship with age and mortality. Very clear. If you're over 80, your mortality goes up to 20%. I'm sorry, part of this slide is cut off. I'm not sure why. But you can see that we have essentially no mortality in very young people, and we have 1 in 5 mortality in the oldest people. And then you have a gradient in between. This, by the way, is another big difference between coronavirus and seasonal influenza. Seasonal influenza kills the very old and the very young. Coronavirus does not seem to kill the very young. Okay. So this is an important point. When you try to compare countries and their death rates from coronavirus, you have to adjust for their age structure. Otherwise, the comparison is not apt. Now, since we are talking, I wanted to throw in a discussion of Kuwait. We have been, as Dr. Khaled mentioned earlier, Palladium has been assisting Kuwait in its response and mitigation plan, coming up with the best plan that is likely to have the most impact. And when we do that, we look at the population, we look at Kuwait, like many countries in the Gulf, obviously has what I call a 2-component population. You've got your native Gulf population, and you've got workers. Most of those workers tend to be younger and male, although you have a big chunk of domestic workers that are female as well. But if you look at the age structure of the Kuwaitis in the population, you'll see that you've got 3% over 60. Now, if you compare that to the US, we are 19% above 60. If you compare that to Northern Italy, they are 23% over 60. This gives you a clue as to why there are differences in the outcomes of the epidemic. Similarly, you have essentially people working in a lot of white-collar jobs for the government and in services. And if you compare that to the US, while services is also the largest sector in the US, we have a large population of people who do essential low-wage tasks as well. And this is important for us when we try to understand the impact of coronavirus on the population here. I put this slide in because I wanted to show you, this is fairly typical for the Gulf. This is the age structure by gender, by sex in Kuwait. This is 2018. And this big bulge on the left that you'll see here is actually your external workforce between the ages of 20 and 55. You also have a slight bulge on the right for the domestic workers, but this is the whole population of Kuwait, not just Kuwaitis. In thinking about Kuwait as well, you think about what some of the factors are that will impact the response. How is the health system organized? How is it equipped? How is it staffed? And this we do in every country, by the way. We think about, what are the biggest industries? Where are the biggest risks? How can they safely reopen? That you need to therefore define the profile of the health system and the other sectors within Kuwait. And some of the ways we do that is we look at the number of beds, the number of ICU beds, the number of critical care beds, and we compare those to benchmarks that are usually established by the WHO for countries. And you can see that you actually haven't approached the benchmark for some of these yet, but you've done an excellent job of curtailing critical illness and death. And that means that you're doing some things quite right. And of course, everything can be improved, but there are lessons to be learned that will be useful for other countries in the Kuwait response. I threw this in as well because prior to this pandemic, I mentioned that people like me have been involved in what's called global health security for many, many years. And there is something called a global health security agenda, which is actually supported by the G7 and the G20. And there are many components of the global health security agenda. One of them is to prevent pandemics from emerging from animals. One of them is to diminish the impact of resistance to antibiotics, so antimicrobial resistance. A third is to improve the availability of vaccines to infectious diseases. There are many, many components of the global health security agenda. And we have a way of assessing how countries are doing on their global health security agenda. And in this slide, I'm just giving you a comparison of two countries in the Gulf and one country that is so-called developed, United Kingdom. The first thing I'll point out is it didn't seem to make a difference in this pandemic where you stood on the global health security assessment. So a country like the United Kingdom, which was judged at number 2 out of the whole world, guess who was number 1? The United States. But both the US and the United States have had major issues responding to this epidemic. Whereas Saudi Arabia and Kuwait were judged number 59 and number 47 on this assessment, which was done in 2019. And they have actually done better. But as we said, some of those factors may not have to do with preparation at all. It may have to do with demography and factors within the country that do not impact on the assessment of your global health security preparedness. So that's a lesson for us in the global health security field. It seems that country governance, the ability to rally the entire country and to have a plan that everyone can buy into and respond to, is the most important issue in the coronavirus response. Now, we are still talking about a disease. COVID-19 is a disease. coronavirus is an organism that causes this disease. The specific coronavirus is SARS-CoV-2, a new or novel coronavirus. Since we are talking about health systems and the ability to respond, we've seen many, many examples around the world where different approaches were adopted to dealing with people who are sick. In many countries, the number of people who were sick increased very rapidly. And that led to certain strategies, such as only seeing the people who are very sick and require hospitalization, and sending everyone else home. And this has actually led to more spread. The countries that have been able to exercise more control over the virus took care of every patient with proven infection, even those who did not require hospitalization. They were isolated and looked after in secure facilities. I would characterize this as one of the biggest deficiencies of the American response, where both testing and attention was paid to those requiring hospitalization, and not enough attention was paid to those who didn't. And we were told that they could isolate at home, which is actually relatively hard to do unless you're living alone like I do. So we said earlier that having a good idea of who needs what depends on your ability to assess everyone walking through the door of a health facility to say, you are infected, you have a positive test, but you're not sick. You are infected, but you are sick. I have a plan for both of you. And there are people in between those two categories. These are very important issues when trying to control spread in the community. And when we look at the Kuwait response, we can see that it falls more on the side of let us take care of everyone who is infected to prevent them from passing the virus to people who are not infected. So there are various policies and strategies for dealing with people who are symptomatic. I've put two up here from WHO and CDC. As I said, reality on the ground has mandated that some of this has not been done in the way that it should have been done. 1 second, I'm frozen. I need to unfreeze myself. 1 second. Okay. We don't have a treatment for coronavirus disease, COVID-19. What we have been doing, and by the way, this applies to the whole world, what we have been doing is what's called symptomatic treatment or supportive care. And that includes, of course, supporting the respiratory system, should it be compromised, supporting any other system, should those be compromised, and keeping patients hydrated and with good nutrition until their own bodies can help them to recover. One key point that I'd like to leave you with is that while the respiratory system or the pulmonary system is the most affected in coronavirus, it can affect every system in the body. And we have seen impact on neurology, the kidneys, nephrology, gastrointestinal, musculoskeletal, strokes in the brain, just a huge number of effects. And we still haven't even described the whole clinical syndrome that's possible from coronavirus. And so when we think about treatments, we think, well, what can we do? Well, there's a huge amount of effort going on to understand, can we develop new treatments that don't yet exist, or can we take old treatments that have existed for other viruses and apply them to coronavirus? And there are many, many clinical trials going on of trying to understand do these existing therapies work in coronavirus? And so we have found one that works, which is remdesivir. It works only in a certain proportion of people to reduce the time in ICU, reduces ICU time by about 30%. But I'll talk about that shortly. So essentially, in order to understand if something works in clinical medicine, we normally do a clinical trial. We compare one group with the intervention versus another group without the intervention. And we don't really know who's getting what. That's the best kind of double-blind, placebo-controlled clinical trial. In the context of coronavirus, we don't have the luxury of time to do that. So we've got all kinds of protocols going on, like this one, which is organized by the WHO, which allows hospitals around the world to upload information on treatment for coronavirus that they have undertaken in order to get a clear view of what could possibly work. And I don't know if Kuwait has been participating in this master protocol, but many countries in the world have been. By the way, this is a picture of what coronavirus in the lungs looks like. And as you can see, this is an early picture. You're seeing clear opacities. My cursor is moving over some of those in the lungs. This is a cut through the lungs, a cross-sectional cut using a CAT scan. And here you can see consolidation has progressed. And all this white area, it should be dark. All this white area is in fact not normal lung tissue. This means that once you consolidate that lung tissue, the clinical picture we refer to as pneumonia, this is a COVID-19 pneumonia, it inhibits the ability of the lungs to absorb oxygen and release carbon dioxide. So the master protocol is giving us new information. There are also specific protocols, one of which I mentioned is remdesivir. Remdesivir is a drug that was developed for Ebola. It is what we call a nucleoside analog. By the way, some of the drugs we use to treat HIV are nucleoside analogs. So we have information that we've used in other diseases. Some of these drugs have activity against multiple types of viruses. And as I said, one phase 3 trial has determined that remdesivir reduces critical illness by 30% and was granted what we call emergency use authorization in the US for use. Here are some of the other drugs that are currently being tried for coronavirus in various clinical trials. Most of these are classical antiretrovirals. Some of them are used in SARS, some of them are used in MERS, some of them are used for HIV. We're trying them all in order to understand what could possibly work. So the treatment summary is we do not have a specific treatment. We must support the patients. Those that are eligible for ventilatory support are the ones where we get most of the mortality. In some hospitals in the US, people who go on ventilators for COVID-19, 50% of them died early on. And in fact, that's one of the lessons we learn is that we actually have changed the criteria for putting people on ventilators because of that high mortality. And we're trying to preserve ventilatory support for people who absolutely are not responding to any other intervention. I'm just debating whether we should talk more about treatment. Obviously, any reasonably well-equipped hospital can treat coronavirus patients. Having ICU beds available is very important for those that need ventilatory support. And having the ability to maintain nutrition and hydration is also very important. We found in the US where over 50% of our deaths have been in those over 70 years old, over 65 actually, that many of them came relatively poorly nourished and systems shut down for other reasons than coronavirus. Having said what, I'm giving you a lot of information, but I'm still telling the story. The story is now at the point of how do we know what's going on around us? We talked about reported cases, we talked about reported deaths, but frankly, those are, we said, the tip of the iceberg. One of the key questions we need to ask ourselves is the numbers of reported cases, 6 million cases around the world today, with a mortality of 370,000. That implies to me that we have many, many more circulating cases than we know about. As we said, if we had a mortality rate of 1%, you essentially got 36 million cases around the world, of which we've detected 6. And so to understand how to do better around that, we need to understand the natural history of COVID-19 and coronavirus. And so we need to relate the clinical picture and the lab picture with the epidemiological picture. And this is also the basis, Dr. Khalid, of the models that we use, the SIR models, which is based on the number of people who are susceptible to an infectious agent, the number of people who are ill, and the number of people who have recovered. Now, when we say recovered in this case, recovery in an epidemiological standpoint includes death, okay? So really, it's not recovery, it's resolution.
Dr. Farley, if we can spend, sorry to interrupt, if you can spend the last 2 minutes to wrap up, since time is almost running out, and we have 3 questions.
Certainly.
Thank you.
I'll just take 2 more minutes. So just keep in mind that we are trying to get a much better picture of what's happening out there. And one of the key tools that we'll be using is not just antigen tests to detect the virus in someone, but antibody tests to detect those who have been exposed and who are potentially immune. And potentially immune people are very important in a recovery plan. If much of your population is potentially immune, and I use the word potential for a good reason, we can discuss. If much of your population is potentially immune, then your ability to have confidence in a reopening plan is greater, okay? This is just a cartoon showing how the antibodies evolve. There are 2 types, and the one that you're left with that is protective and the basis for most vaccines is IgG. I think I will stop there. We have been helping Kuwait with its response and mitigation strategies. None of this is a secret. There are no magic bullets. And the most important thing is having a plan. Even if your plan is not 100% accurate, your plan can be modified. And countries without a plan are going to suffer greater consequences. I'll stop there. Thank you.
Thank you, Dr. Farley. So we'll take the first question from Mr. Frank. Is there an anticipated second outbreak or second wave of outbreak other than the potential associated with recovery?
Yes. Thank you. That is a very important question. And again, we have to look at our sum total of knowledge about this virus. So let's ask ourselves a couple of questions about that question. One is, do people who recover from having COVID-19, do they have protection from subsequent reinfection? I would say the answer to that is, while we have a couple of cases that appear to look like that, on further examination, they actually don't have productive infection subsequently. So if you have COVID and you recover, you are protected. That is my prediction. How long that protection lasts, I don't know. So having more and more immune people in your population will go a long way to prevent the second wave that we're talking about. Now, what is the second wave? Well, if you think about 1918, the flu, we had a larger second wave than the first wave. So that is an important historical lesson. Is this something we can say is going to happen or will not happen with coronavirus? We can't say for certain, But what we can say is that so many countries are in different stages of the epidemic that there's absolutely no reason why the virus can't circle back to a place that already had their initial epidemic. Our best knowledge about antibodies and protection is that even in places with huge epidemics like New York, like France, like Spain, like Italy, even in those places, only 5 to 10% of the population showed positive antibodies in surveys. So 90% of the population is still susceptible. So can the virus circle back to New York, circle back to Italy, circle back to France? The answer is yes.
Excellent, thank you. So second question from Mr. Hamid. What was the shortcomings of Kuwait plan for the pandemic?
Well, I'll have to ask Dr. Khalid's permission. I would say there were no specific shortcomings to Kuwait. Like many countries in the Gulf, Kuwait took very important precautionary measures. I would say the dawn to dusk, well, the absolute curfew in Saudi Arabia is probably the most extreme example of the lockdown in the region. Kuwait's lockdown was not absolute, but it was enough. And as I said, even before the lockdown, Kuwait was doing relatively well in terms of testing and in terms of supporting patients in hospital. You've kept your mortality rate very low. Some of that is related to age. So are there things that can be improved in the Kuwait response? Absolutely. And that is what Dr. Khalid and his team is working on. He's got lots of good advice, not just from us, but from lots of other people. He's trying to take the best information as all of this information is emerging so quickly. How do you capture it and distill it and make sense of it? That is what a good planning or a good plan should involve.
Excellent. Third question from Mr. Ella. In the beginning of the presentation, a slide containing a timeline for the most famous pandemics from the 1800s until now shows that the gap between pandemics was big, almost 150, 200 years. But in the last 20 years, the gap is shrinking significantly, where we had almost 5 major outbreaks from 2000 until now. Do you think we're on the verge of another pandemic after COVID-19 in the short years to come?
Yeah, that is an excellent question. And as I pointed out earlier, when I talked about those slides, the time between significant global epidemics / pandemics is shortening. And I brought up 3 reasons for that. One is, of course, the increasing human population in dense settlements around the world. The second is increasing human travel to all parts of the world in a very short time. And the third was the opportunities for interaction between humans and animals. All of those are increasing. So my prediction is yes, since we don't have a very good idea about all of the viruses and other organisms that exist in animal populations, like bats, which cause no impact on them. But we haven't done full surveys of viruses from every animal species. So the notion that this can happen again is, of course, it can happen again and it will happen again. And it's likely to happen relatively soon rather than in the distant future.
Excellent. Dr. Tariq, I see your question, but we're out of time, so I apologize for that. Dr. Farley, thank you so much for an excellent presentation and a deep dive into managing COVID-19. Thank you for our dear audience, and we look forward to seeing you hopefully next month. Thank you, everyone.
It's been my pleasure. Thank you very much.
Thank you very much, Dr. Farley, for this wonderful presentation, which is very engaging, by the way. And my apology for all the other questions, but we are running out of time, and we would like to keep the time as being mentioned in the invitation. Thank you very much, Dr. Farley. Thank you.
Thank you.